All right, so this lecture is an introduction to eukaryotes, the eukaryotic organisms, our third and final domain, but it's going to take the rest of the semester for us to get well acquainted with the eukaryotes. We'll start out by talking about protists, getting into our diversity of the eukarya. So, what is so great about being a eukaryote? Well, first and foremost, eukaryotes get to have sex. That's pretty exciting, although maybe not for the reasons you're accustomed to thinking about that being exciting. Another thing that's great about being a eukaryote is you get to have complex organelles like mitochondria and nuclei and endoplasmic reticulum and all your membrane-bound organelles. In addition, many eukaryotes also have chloroplasts. So that's three things right there that make being a eukaryote great. So having mitochondria enables us to produce lots and lots of ATP so that we can become truly multicellular. Occasionally also, we get to be multicellular, as you can see here, because we just have so much ATP leaking out all over the place that we just don't know what to do with it. So we established tissues and tissue systems and all kinds of other ways of being multicellular. Things that prokaryotes, bacteria and archaea, cannot exist. What else do we find in the eukaryotes? What are some shared derived characteristics, some synapomorphies that we see in all eukaryotes? Well, cells with nuclei, that's something that we use as the primary defining feature of eukaryotes. In addition, we have other membrane-bound organelles, like our Golgi bodies, our endoplasmic reticulum, vacuoles, things like that. We have different types of sexual life cycles. Chromosomes are typically linear and compartmentalized into some number that's more than just one chromosome. And then other things like having a well-defined cytoskeleton with microtubules and microfilaments and intermediate filaments and things like that. So, what meiosis? Yes. When we talk about sex in eukaryotes, in the very broad sense, we're talking about alternating between meiosis and I wanted to say fermentation, but what I mean is fertilization, and mitosis in between. So we're truly exhibiting mitosis and meiosis, and adding in fertilization. So what is up with meiosis? So what happens in meiosis is we go from having one set of chromosomes to having two sets, so haploid to diploid through fertilization, and going from diploid to haploid through meiosis. So organisms vary whether they are most frequently found in the haploid state or the diploid state, and they also vary in when they grow by mitosis. So this gives us three possible scenarios, and here they are. So animals, like you and I, exist as diploid organisms. We started out as a zygote. The zygote is the first diploid cell. The zygote divides by mitosis to form a multicellular organism that's diploid, like this mouse or a jellyfish, or my dog that I'm petting right now, or the cat that's sitting in my lap. All animals are diploid until they want to reproduce, and then diploid cells go through meiosis as they do, going from diploid to haploid, and those haploid cells in animals are the gametes. So the gametes are the haploid cells. They don't go through mitosis anymore after they become gametes. The only route for them to continue to survive is to fuse through fertilization and form a new diploid cell, a new zygote. So that's animals. But that's not the way it is for all eukaryotes. In some organisms, like fungi, they are predominantly haploid. they spend their life as a haploid organism, which means they are always haploid except when the gametes fuse in fertilization to form a zygote. The zygote doesn't divide by mitosis like we do, and undergoes meiosis immediately and forms haploid cells again. These can divide by mitosis to form a haploid organism, and then the gametes are formed not by meiosis, like they were in animals, but by mitosis. All right, so the third flavor, or the third type of life cycle, alternating between meiosis and fertilization, is that we have mitosis occurring on both sides of the life cycle. So we have a haploid organism, and we have a diploid organism. So if we start with fertilization, we start with our first diploid cell, the zygote. That can divide by mitosis to form a diploid organism. Meiosis can occur to form a spore, is usually what ends up happening. The spore can divide by mitosis to form a haploid organism. And that haploid organism can then produce gametes by mitosis. So both diploid and haploid individuals in plants, some algae. So 3 different types of life cycles. So the first case, evolutionarily speaking, was zygotic meiosis, where the zygote undergoes meiosis. Zygote undergoes meiosis. When you were a zygote, you did not undergo meiosis. You underwent mitosis to form the you that you are. Evolutionarily speaking, gametic meiosis evolved second, with a diploid organism forming from the zygote and gametes being produced by meiosis and fusing with no further mitosis to form a zygote again after fertilization. That's gametic meiosis. And the third type is called alternation of generations, most frequently, but we could also call it sporic meiosis because a spore is formed by meiosis. See, here's meiosis, and here are the spores. So we see a number of trends besides just these different life cycles that occur throughout the eukaryote, the domain of the eukaryotes. So I've listed them here in order from ancestral, the ancestral state, the plesiomorphic state, to the apomorphic or the derived state. This is how it used to be. This is what came along later. And I'll go through this list one by one. So going from unicellular to colonial to multicellular. So in unicellular organisms, like this unicellular prokaryote, you can see this is the entire organism right here. When it divides by mitosis, it forms 2 individuals. It has reproduced. In colonial organisms, here are a couple of different green algae. Here's Hydrodictyon. Here's Pediastrum. You can see there are multiple cells together, but all of the cells look more or less the same. They don't have division of labor. They don't form tissues or organ systems or anything complicated like that. Just one type of cell. All these cells, they do work together, but each of these individual units is more or less the same. So you can see all of these little Pediastrum cells look pretty much identical to each other. Same with these Hydrodictyon cells. They all look more or less the same. And if we took one of these cells out, there's a good chance that it could reform a new colony just by cell division. Now, here's an example of a protist that is truly multicellular. Here's a brown alga. Here's a diagram of it. Here's a kelp, a photograph of it with some other multicellular organisms, these fish found in this kelp forest. And you can see that here we have division of labor. We have different types of tissues and organs, and this is true multicellularity. And you may wonder, how did it all get started? How did multicellularity first develop? Well, from a colonial organism. And as it turns out, we hypothesize that multicellularity began by a few cells becoming specialized. And these were reproductive cells. So reproductive cells were the first way of dividing labor between different types of tissues. So that's kind of interesting, you might think. I do. As far as the gametes are concerned, we see an evolutionary trend going from isogamy to anisogamy to oogamy. This is an oogamy, this is oogamy. O-O. So we say both of those O's. So in isogamy, the gametes look the same. So these two things labeled here as B are gametes, and you can see there's no way to tell them apart. They look like they're the same. Chances are there might be some chemical mechanism to prevent them from fertilizing their own kind, but they're the same shape, they're the same size, no real difference. So iso, the same, gametes. From there, we see a differentiation. We see microgametes and macrogametes, small gametes and big gametes, and these structures labeled C here. This is anisogamy. And in anisogamy, well, that's what you see. You can tell the difference between these two gametes. These are going to fuse together, and we can tell that because one's micro and one's macro. Eventually, we get oogamy. OO is a prefix that means egg. So here we have a large, typically non-motile gamete, which is like the egg. And we see a smaller motile gamete, which is akin to the sperm. So we go from having the gametes looking the same to being a little bit different in size to being very conspicuously different from each other. So they're different in their shape even. We see in organisms that go through alternation of generations, through sporic meiosis, they go from being isomorphic, or the same in appearance, to being different in appearance, or heteromorphic. So here are a couple of different green algae. This is sea lettuce in the genus Ulva. Here's the diploid form, which can go through meiosis to produce a haploid form, which can, after producing gametes, go through syngamy, or fertilization, to produce the diploid stage again. You can see it's kind of hard to tell these two things apart because they are the same shape. They look the same. Isomorphic. The only way to tell diploid ulva from haploid ulva is to just sit and wait and see which one's going to produce gametes and which one's going to undergo meiosis. Otherwise, they are isomorphic, or the same in appearance. As opposed to Derbezia here, which you can see has a very different diploid form from the haploid form. So these little characters here represent meiosis and fertilization. So here's the diploid stage, which looks very different from the haploid stage. As far as the dominant stage is concerned in alternation of generations, we see an evolutionary trend from being gametophyte dominant, or having the haploid stage being dominant, to being sporophyte dominant, which means the diploid stage becomes dominant. So this is something we only see in alternation of generations, and we're going to really see a lot of this when we get to plants very shortly. So I'm not going to talk too much more about it now. So this is just kind of foreshadowing of that happy day. So talking about the protists, and once upon a time we talked about the kingdom Protista. It wasn't really a very good kingdom in the sense that it's not considered to be holophyletic. It's not containing all of and only the descendants of a single common ancestor. It's kind of been treated as a dumping ground, or if we didn't know where to put it otherwise, we would put something in the kingdom Protista if we knew it was a eukaryote. So here's Jeff Foxworthy, and he's telling us, you might be a protist if you're a